Thank you for the invitation. It's a great pleasure. Indeed, this talk I'm going to focus on my recent research this couple of years, especially since 2006. And the work is from ground up. It's from very basic preparation of animals. So I learned the process, worked as a graduate student or undergraduate student even, all the way up. And this is, so pretty much the results you see now is up to last year, right around this time, summertime actually, late summertime, it's data become reasonably useful. So we started, finally we get to the point of able to generate some results. And this is what you're going to see. And so I'm going to talk about, this workshop is in the context of computing, intelligent computing. So I'm going to talk about computing with spikes. This is, to begin with, let me show you a video. I suspect this is going to happen, so I have the video here. This is a sequence of about 12, 13 seconds, and this is actually from the cortex of a rat. This is from my lab, and I recorded this signal. I did the surgery myself, recording the signal. So what you see is there are 14, no, there are 16. Okay. There are 16 channels, and this is one of the channels. So these are the spikes, are what I mean by neural spikes. So this is the fundamental computing paradigm or the signaling mechanism inside the brain, inside the rat's brain, inside our brain. So the fundamental question is, how do we compute? How does the brain compute using these spikes? And what you saw here is just one sequence. And for one sequence, over the years, neuroscientists have worked extensively on understanding a single neuron, the firing mechanism, and the inputs and the outputs, the axons, and cellular, molecular, and ionic level transportation, and variations, noise, and so on and so forth. So people have a reasonably good understanding of how a single neuron fire, how a single neuron spike is generated. But however, you have, in the brain we know we have billions of neurons. When we just a twitch of a finger, that fires a firestorm, that brings up a firestorm in the brain. That is, a lot of spikes are computing, lots of neurons are participating. How a network of neurons compute, this is a huge challenge. So the idea of cracking the neural code is the thesis of many scientists trying to work on, have been focusing to try to resolve. And from a pragmatic engineering perspective, let's say, can we decipher the syntax of the set of rules that transform electrical pulses, the ones you saw just now? Can we derive a set of rules that govern the brain? The brain fires according to this syntax, according to these rules, and that relates to our behavior, our thoughtful, conscious act. I make my decision. I have this happy feeling or sad feeling, or I get excited, or I do so and so. So what is behind all of it? Is it possible to decipher such rules? And this is a very difficult question, let alone every single person is different. Rats are very different. That's what we saw in the laboratory. They're just like people. They have personality. Some are faster learners, some are slower, some are happier, some are easy to cope with, some are more social, some are less social. So even rats are giving us so much variations of personalities and decision-making strategies. This is what we observe as well. They learn things differently. They use different strategies. So is there a possibility to decipher such set of rules? Before we go that far, before we can answer that big question that everybody's trying to answer, we're still way far from the answer, getting the answer. We try to, in my laboratory, I try to provide some basic fundamental studies, create some fundamental frameworks, so we can provide some mechanistic support to the understanding of the frontal cortex, not the entire brain, but part of the brain. And this part of the brain is associated with, recognized for cognitive decision-making, higher level information processing. When we try to make conscious decisions, when we try to pay attention, when we do things with working memory, for example. This is the area where we identify. So again, going back, now we have a focus area to probe and to study the code. And generally speaking, when people talk about coding, computing with spikes, many years ago, the rate code is one of the most commonly used. And the rate code, simply put, is just you count a certain length of time, and you count the spikes in this time window. And then it's basically the unit time, spikes per unit time. That's the rate. And that it's very handy. It's basically a first order statistic, and neuroscientists have been using that quite successfully, explaining some of the questions relating to how you decode from spikes to behavior. So this, on the other hand, in the late 80s, early 90s, scientists in the laboratory starting to realize there is the spike timings also bring out lots of good information. Synchronization, spike trains from different neurons, they actually tend to synchronize under some specific stimulus conditions. And recent 10, 20 years, and in between there is all sorts of hypotheses, computational paradigms, and modeling schemes. And people try to analyze the interspike interval, trying to study the spike train in the frequency domain, try to do correlation analysis, and so on and so forth, information measure. So this is all these efforts are aiming at trying to reveal the fundamental neurocoding, the neurocoding paradigm inside the brain. And so as I implied just now, there is no, right now we're far from a good understanding of the global neural perspective and its relation to behavior. But there are some basic principles that neuroscientists start to agree on, one of which is individual neurons, they represent relevant features of the environment. And to encode accurately a piece of information, a population of neurons are involved. And many of these studies are using external stimulus. This is especially in the auditory cortex and in the visual cortex. Other things like cognitive function are less investigated, on the other hand. So this is a big picture of where things are in terms of understanding the code. So now I'm trying to get into more specifics about the code. And first of all, it's the rate code. And as I said just now, the rate code is basically a count of average number of spikes or action potentials per unit time. This rate code alone is insufficient to account for many sophisticated spike trains, especially from a population of neurons. The rate code, however, in motor neurons, not motor cortical neurons, these are motor neurons activating muscles and so on. The motor neurons are the strength of innervated muscle flexion, muscle activation, actually depends solely on the firing rate. So this is something clean, nice, and the firing rate alone can explain pretty well. But it's not the cortical neurons. Things are not much more complex in the cortical neurons. And here's another example of the temporal code. This is a famous experiment from Gray's lab published in 1989. And this is basically indicating the stimulus synchronous oscillation from neurons when a visual stimulus was presented to the monkey. So they hypothesized the oscillatory responses. They may provide a general mechanism for the neural waveforms to propagate from one brain region to another. And this provides a temporal coordination of these different brain areas. So this is so the rate code and the temporal code, these are, I say, probably we call this two extremes of neural coding, and in between there is a lot more. And these coding schemes have explained certain things, but not everything. And so to quickly summarize, a very high-level summary, in up to this state, the sensory systems, it is generally accepted. The firing rate is correlated with the intensity of the stimulus. This is especially referring to auditory cortex, for example, the visual cortex, for example. And rapid changing stimuli tend to generate precisely timed or synchronized spike firing. So these conclusions are reasonably established, reasonably accepted for sensory systems. But how neurons encode information when it comes to intrinsic brain states? Intrinsic brain states, we mean the thought process, for example. You try to explore the environment, and you try to learn to do something. When you try to derive a command, and you steer your car. At a certain point, you feel like you need to speed up. At a certain point, you feel like you need to go to the right lane. How this process, where you don't have a clear external stimulus, say, externally nobody says, now you need to make the turn, now you need to speed up. Nobody does that, but your brain somehow kicking this process, and it has an intrinsic development, and how these intrinsic developments take place. And these are even harder questions than the stimulus-driven spike coding. And this type of questions, when we talk about intrinsic state, these are usually related to perception, to cognition, to decision and control. This is usually involved in the frontal cortex. And the frontal cortex basically is here. And the frontal cortex is probably one of the most convoluted, most complex area of the brain. It has many different types of cells, much more complex than the deeper region of the brain. So with all this said, is it still possible to try to decipher the general spike computing principles? So as I said, under this very challenging problem formulation, under this very broad, almost elusive objective, and what people have done is to bring this problem down to a specific task domain. into specific neural cortical areas. And then they try to design experiment, and then they try to understand this part of the brain and try to understand this part of the neural coding within this part of the brain. So in my laboratory, what I do is I use a rat model, that's what I mentioned just now, and I use natural behavior. Natural behavior meaning the rats are unrestricted, they do what they want. Of course, they're inside a box, they can't run everywhere. And they're freely moving. The reason I say this is many neuroscience experiments, the animals are restricted, the arms are not moving, the legs are not moving, even the head is not moving. These are all very stereotypical behaviors. So I try to relax that condition. And the cognitive task I ask the rats to do is a decision control task, as I'm going to talk about in a little bit. And in terms of probing the brain, I use electrophysiology, which does single unit recording. Basically, what you saw just now are single units. Single units meaning signals from a single neuron. These are actually from single neuron in the brain. In theory, we can do this on humans as well, but nobody is willing to donate a brain for a live person would let us to do that. So that's why the rat model is very helpful in this regard. And another important aspect is computation with neural spikes and the modeling. And the end goal is we try to correlate the neural spikes from many neurons to intelligent behavior of an animal model in this case. And this is a pretty challenging setup. Usually, as I mentioned just now, neurophysiologists, they do experiment with somewhat stereotypical experiment with restricted movements, with restricted activities. And behaving animals are psychophysical experiments where psychologists use. And of course, they don't do electrophysiology. So this platform is quite challenging, takes time to build, and takes patience to tune and for data to come out. So here's the experiment. The rat is inside a Skinner box, and he faces a panel, and this control panel has lights on the side and has two control panels underneath. And if he sees the left side, if he sees a cue on the left side, he should go press the left pedal. And if the right light goes on, he should go press the right pedal. So this is a association. This is an association task. And the rat didn't know. Before he went into the box, he had no idea what he was supposed to do. So he learns, and also there's a center pedal. There's a center cue, I'm sorry. There's no center pedal, there's a center cue. So there is a center cue. And if the center cue turns on, he's supposed to do nothing. He's supposed to not to press the left nor the right. He'll just wait there. And if he waits sufficiently long, he picks up his reward. This is in behavioral study, this is called inhibition control. And inhibition control is important from the cortex. And many times, the lack of inhibition control is identified with aging. So for example, for aged folks, it's inhibition control actually is a test to see how capable your brain is as you age. So this is not that easy of a task. Even for human, it's not as simple as you thought. So the rat went in from the naive state, and the rat learns to perform this task. And we record the process of his behavior. He learns to do this task. His accuracy was very low initially, and then gradually he improves. And as he learned to perform this task, many trials, and we record his neural signals at the same time, and then we come back and analyze his neural signals. So that's basically the process. And the electrode is implanted, as you can see, the electrode is implanted in this area, basically covers a big chunk of his frontal cortex. So this is a behavioral learning curve. We summarize these. This is a summary. This is a summary of behavioral learning curves for many rats, just for the ease of displaying the typical behavior. So basically, what you see is this is day over day. It typically takes about 2 weeks, 3 weeks. Some faster ones, we have seen them learn in 3 days. Some slow ones takes over a month or two. Still can't figure out. And the vertical axis is accuracy on the task I just described. So the rat learns to perform this task. Initially, they were all naive. They didn't know what they were supposed to do, didn't know what the light means, so they gradually learn, because if they do it right, they get a food reward. So they get incentive, they keep working until they get very high percentage. So usually, they learn up to 85, sometimes 90, and kept it there for a few days. And we usually keep the rats working for as long as they can, but the physical condition changes. Sometimes the head cap came off, and other reasons, and then we have to sacrifice the rats. So these are the typical learning curves. And here's what we call this is very common in neuroscience, this is called spike raster. The spike rasters, each line of these dots is one spike train. So these are each line is one trial, one spike train. So another trial, a spike train from another trial, repeated trials. So these are repeated trials. This is each one of these dots is what you saw just now at the beginning when I showed the video, where you see the spikes. This is basically each dot is a spike. So this gives you a snapshot. This gives you a snapshot over the time course of a trial, how each neuron respond. So collectively, statistically, it gives you a profile how neurons, what typical characteristics are of a neuron's response to this task behavior, starting from the cue light is on to the point where he picks up his food reward. This is a typical, give you an idea, intuitive idea what it looks like. So next, I'm going to show you 3 results that we just concluded, we finished. 3 interesting results. One is based on the rate, firing rate. The other one is based on spike timing. And another one is based on network modeling. This is something there is a lot more potential to explore. So the question for the rate coding is there anything we see just from the basic spike rate, just by computing the rate, what we see from the rat, that we can relate to his behavior? What we notice after computing all these spike trains, we realize the spike rate, high or low, are related to trial outcomes. The trial is correct or the trial is incorrect. And the rate modulates over the course of a trial, from the start, when the cue is on, to the point when he picks up his reward. The neuron fires, the rate changes. It's not always a constant rate over a trial. And also, the rate modulation occurs differently for different phases of learning. When he was learning the task to the point where he finally learned the task, these 2 phases, the firing rates are different ones as well. So basically what we saw is firing rate changes over the entire process of learning and within each trial of his behavior. And this is a behavioral data summary where it says, so we divided the data into 3 stages. This is the naive state where he basically had no idea what he was supposed to do. And then he starts to make sense. Okay, this is left light, left press. And this is learn. He learned the task. Okay, he totally understands. He makes very high accuracy, makes very few mistakes. And this is totally behavioral trials. And we see this type of learning in psychophysical experiments as well from other people. And what's interesting here is if you focus on the learning stage, as you can see during the naive stage and during the learn stage, it's statistically you don't see a clear difference between you don't see a clear difference of error correction. This one you do see error correction. Error correction meaning I have two trials. If the previous trial I make a mistake, then I realize I made a mistake. I better think about my mistake, analyze it. And the next trial I make corrections. And this is shown here. You see the red is post success. The black is post error. So after the error here, you see the post error, the rat is paying attention. So his accuracy is actually increasing over the previous trial's accuracy. So this basically shows the rat is capable of picking up from his own mistakes. So this is behavioral data. And what about neural? What is reflection in the neural data? And the neural data shows, if I look at the, these are incorrect trials. So we have many rosters, as I showed you just now. So all the blue ones are incorrect trials. The green ones are the correct trials. So if I look at these two types of trials, if I look at the current, just I'm doing the current trial, and during the reward stage, I'm expecting a reward. Do I get a reward or I don't get a reward? You see his expectations are different. This is when I'm only looking at the current trial. I'm not considering correlation with the previous trials. So then he realizes the difference if he does it right or he does it wrong. But if you look at the previous trial outcome, as you can see, the previous trial, again, blue is incorrect, green is correct. So the trial, if the previous trial is incorrect or the previous trial is correct, the firing rate is very different. This is firing rate over the time where he was executing the current trial. So if I made a mistake during the previous trial, I modulate my brain activity so that this current trial, I do something different. And this is actually shown, the rate is shown here. You are actually, the rat actually is modulating his brain activity. You see this reflected in the rate. And this is a very coarse measure of the brain activity, but it does show this activity is very different. So that's what our, that was the first key observation we made. So basically, rate alone already reflected something quite significant from the rat, where this data have not been shown before, where mostly people have been showing psychophysical experiments. So the neural data, actually, we have seen this great difference. And the next thing I like to show is spike timing. Is there anything in relation to spike timing in the course of a trial, and also before learning and after learning the task? So here is a computation where the x-axis is one spike train, and the y-axis is another spike train. So basically what this goes on is we try to compute the correlation, the overlaps between the two spike trains. And here you see stronger overlaps, then the color is brighter. And here you see less, you see less overlaps between the trials. So basically, along the diagonal, meaning the time alignment is closer, and the off diagonal, the time alignment is far off. So if the time alignment is close or identical, that means, and the color is bright, that means the two spike trains has strong overlaps. And this figure shows these two neurons actually are strongly correlated. They tend to fire together during the initial period of this trial. So this trial in the initial period, and they tend to fire together, not in the later stage. So when he was paying attention, he was trying to execute the task. The two neurons actually are more aligned. That's what this result is showing. So this one shows before learning, after learning. This is during learning, this is after learning. So this shows during learning, he is actually, there's more timing correlation, they're more aligned than after learning. So this figure is basically summarizes what I just said. During learning, initially, he's paying more attention. The spikes are more aligned than than the later phase. And during learning, the alignment is stronger than after learning. So this is a summary of one rat, and we see this in many rats. So these are simple observations from rate code and from time code. And the next question, we try to go a little bit more complex approach, where we do a network, a model-based approach. And this is something we're thinking from because this is a learning task, and we're thinking the problem of spike timing-dependent plasticity. And this is of significance because learning has been hypothesized as taking place in terms of spike timing-dependent plasticity has been hypothesized as a mechanism of learning, where basically if you have two spikes, the input spike and the output spike are closely, if the input spike leads to the firing of an output spike, then the synapse tends to become stronger. And this has been done in vitro. Basically, you have brain slice. You do this in vitro. But in vivo, you won't be able to verify this because you can't. In vitro, you cut the slice, you go to the two neurons, you measure if they fire one early, one late. But in vivo, you won't be able to do this. So we are proposing, this is a pretty wild hypothesis. We are trying to, this is the line of thinking we're putting together right now. So this provides, probably this model-based approach can provide us with a way to interpret in vivo learning of the interaction. So the basic idea is I have one neuron, I have this spike train, multiple neurons, and what is the probability of firing a spike? So this is a predictive model now. This is, I cut all the slides on the model derivation, model justification verification, and so on. This is a big neural network modeling, computational neural network, I mean. There's a big section on computational neural network modeling. The idea is we want to predict if there is a spike in the next time segment. So this problem is we formulate it as a, so there is a whole argument of point process and inhomogeneous Poisson, identifying the arrival rate parameter, so on and so forth. I'm cutting all this back. The idea now is how we can estimate the interactions between two neurons, I and C, during time M. How we can estimate this coefficient. This basically measures the strength of the two neurons interacting. So this model, of course, this model is a function, I call it a functional model. And using this functional model, we try to provide an argument that This is spike timing dependent plasticity in vivo because we have this model, because we can use help from this functional model. And this functional model is not a physiological model. This is not a Hodgkin-Huxley. However, this is, again, this is cut out from this because we have limited time. This is based on the spike response model. The spike response model is validated against the Hodgkin-Huxley. And this functional model, we try to fit through our data. And there is a whole set of procedures. There is also maximum likelihood and so on and so forth. And we have this, basically it's a least square solution. And estimating the interaction coefficient. And here's the result. So our goal was to, based on that network model, we try to estimate the functional interaction strength between every two neurons. And the result is quite interesting. So initially, before learning, the interaction strength are shown. Some are hot, some are cold. Hot meaning positive interaction, very strong. And after learning, the color cools down. It seems the interactions have become focused onto some neurons. And this is evidenced by many rats. And more, as you can see, the percentages is higher for inhibitory interconnection coefficient. So basically what this seems to be, the using this functional model, using in vivo data, the indication seems to be that through learning, the neurons, the functional interactions seem to be cooling down, seems to be becoming more and more selective. Some neurons, the connections become more connected, while others are cooling off. So this result, I like to talk about the last bullet and then I'm done. So this result is we talked about rate coding, we talked about temporal coding, and also the network model coding. And this is a place where this is obviously a place with big data, science of big data. Every day in my laboratory, for one session, I record a couple of gigabytes, sometimes about 10 gigabytes of neural data. And from this 10 gigabytes of neural data, we go sort the data, sort the spike, and then we start modeling, we start computation. So this is we're scratching the surface of the neurophysiology of neuroscience, and many this type of experiments are needed before we can actually make sense of how the brain actually code behavior. This requires simultaneously, this requires lots of good experiments, requires lots of good technology, because probing the brain, after all, is not something easy. It's for a good year and a half, I didn't get a single byte of data. It's very difficult because of a head stage problem. The hookup is just not good. So this requires good technology, requires good design experiment, and also, as you saw, you got a glimpse of it just now, it also requires good modeling, predictive model, to interpret what we cannot do in the experiment. So I would like to summarize, sum it up right here.